Embryonic development is an intricate and fascinating process that involves the growth and differentiation of various organ systems. One such organ, the spleen, plays a crucial role in immune function, hematopoiesis, and homeostasis. Understanding the complex events that underlie the development of the spleen is essential for comprehending its structure, function, and associated pathologies. During development, the spleen receives its blood supply from the dorsal aorta. The splenic artery arises from the celiac trunk and follows a complex branching pattern to supply the spleen. This branching is necessary for adequate blood flow and oxygenation. Development of the splenic artery is influenced by factors such as SPT4, FGFs, and SHH. Angiogenesis, the formation of new blood vessels, is a complex process that plays a crucial role in spleen organogenesis. During embryonic development, angiogenesis is tightly regulated by a network of pro-angiogenic and anti-angiogenic factors. Within the developing spleen, the process of angiogenesis is initiated by the release of pro-angiogenic factors, such as vascular endothelial growth factor, VEGF, and angiopoietin-like 2, UNCL2. As angiogenesis progresses, endothelial cells migrate and proliferate, forming primordial vascular networks within the developing spleen. These networks are further remodeled and refined through the pruning and stabilization of vessel branches. Notably, the process of angiogenesis is intimately linked with the establishment of cellular niches within the spleen, which are essential for proper hematopoiesis and immune function. In summary, during the development of the spleen, splenic cords and sinusoids form. Splenic cords consist of reticular cells and macrophages, providing structural support and supporting hematopoiesis. Sinusoids are specialized blood vessels that allow for blood filtration and exchange with surrounding tissue. In summary, the development of the spleen involves intricate interactions between mesenchymal and epithelial cells. These interactions are guided by molecular players and signaling pathways, including the WNT, BMP, and FGF pathways, which regulate cellular differentiation, growth, and patterning of the spleen. Hematopoietic stem cell, HSC, homing and engraftment in the spleen is vital for the development and function of the blood system. HSCs arise from specific cell types and travel to the spleen through the bloodstream. In the spleen, HSCs interact with different cells and molecules to ensure their proper integration and differentiation into various blood cell types. The splenic microenvironment provides the necessary signals and support for HSC maintenance and differentiation. The spleen plays a crucial role in the development and maturation of distinct populations of lymphoid cells. Lymphoid progenitor cells migrate and differentiate within the spleen, guided by developmental cues and signaling pathways. Notably, the Notch signaling pathway promotes the generation of functional B cells and T cells. Additionally, the spleen provides a microenvironment for the maturation and education of B cells, where they undergo processes like somatic hypermutation and affinity maturation to acquire high affinity for antigens, enabling effective immune responses. This video section discusses the development of the splenic stroma, which is composed of different types of cells. These cells provide structural support, regulate blood cell production, and facilitate communication between immune cells in the spleen. Cytokines and growth factors control the development and function of various cells in the spleen. They guide cell fate decisions and promote maturation and differentiation. EELs, TNF-alpha, CSFs, and EPO are key players in this process, influencing immune and hematopoietic cell types. Proper timing and location of their expression are vital for successful splenic cellular maturation. The spleen plays an important role in extramedullary hematopoiesis, where blood cells are produced outside the bone marrow. Factors such as cytokines and immune cell interactions regulate this process. This knowledge is crucial for understanding and treating hematological disorders. Genetic mutations can have profound effects on spleen morphogenesis, function, and disease predisposition. Numerous genetic disorders have been identified that specifically affect the development and function of the spleen. For instance, mutations in genes encoding key transcription factors, such as PAX7 and HOX11, have been associated with congenital asplenia or functional hyposplenia. These conditions can lead to increased susceptibility to infections and impaired immune responses. Additionally, mutations in genes involved in the regulation of angiogenesis, stromal cell development, and hematopoietic cell differentiation can result in various spleen abnormalities, including hypoplasia or dysplasia. Understanding the genetic basis of spleen development and its associated disorders provides valuable insights into the molecular mechanisms underlying normal spleen function and associated pathologies. Transcription factors control spleen development, regulating gene expression for cell fate, differentiation, and organ structure. Examples include SOX4, GATA3, and FOXN1 for lymphoid cells, and HOX, PAX, and ETS genes for spleen regionalization. Studying these factors is crucial for understanding spleen formation and related diseases. The WNT signaling pathway is important for the development of the spleen. It controls cell growth, tissue pattern formation, and cell specialization. Different molecules and events are involved, and disruptions can cause spleen problems. Studying this process gives us knowledge of how organs develop. In summary, the development of the spleen involves intricate molecular, cellular, and signaling interactions. Understanding its stages, players, and associated pathologies is crucial for comprehending its structure and function. Further research will continue to enhance our knowledge in this fascinating field.